Hello everyone, my name is Selan and I'm a PhD student in Bioinformatics program at Waldebron Research Institute. And in this video, I will shortly present my poster entitled Development of Continuous Protein-Specific Predictors of the Impact of Protein Sequence Variants. Our goal is to predict the impact of variants on protein function, yet this is a difficult problem to solve because it depends on several factors like the genetic component, effect size, location of the variant, and so on. The computational approach for identifying variant effect is an alternative method to complement to the experimental, which are methods developed using different properties of variants to give a prediction with a certain reliability. They are fast and easier to interpret, but given the limitations in their accuracies, cannot be used alone in clinical settings. So, in this study, we focused on two main points. One, predicting the continuous functional changes upon mutation by building protein-specific supervised models. Why? Because continuous predictions contribute to the mechanistic interpretation of the impact of variants. Secondly, considering that the protein-specific predictor pool is limited than the number of proteins, we wanted to explore if a protein-specific predictor can be used for predicting other proteins. To build a protein-specific model, we follow the 5 steps we see in Figure 1. In data retrieval, we collected deep mutational scanning experimental data available in the literature for 29 unique proteins. These assays were then normalized to bring the wild-type score to 1. The number of non-synonymous variants collected for each protein and their normalized distributions can be seen in Figure 2a. Next, we used 10 features for describing our variant's nature, which can be grouped into two sections. Conservation and sequence-based features, calculated from multiple sequence alignment and amino acid replacement information, and structure-based features, calculated from structure predictions obtained from AlphaFold tool. For building the models to handle imbalanced dataset distributions of training sets, we did undersampling to the majority groups through the use of Gaussian mixture models. For making continuous predictions, we used multiple linear regression and random forest regression algorithms. For estimating the performance, different than the standard leave-one-out cross-validation, we applied a more stringent cross-validation method, which splits by position instead of a variant. Lastly, to compare our model's performances, we did comparison against 20 tools in the field. And for the cross-predictions, we used one protein-specific tool for predicting variants in other proteins. Now we move to the results, starting with performance of 29 unique protein-specific predictors. We calculated Spearman's rank and Pearson's values for assessing the performances. Overall, our models worked moderately well for some proteins, including the ones we see in heat maps in Figure 3, TEM1, TP53, and BRCA1. Whereas for others we observed lower performance, including proteins like CALM1 with Spearman rank of 0.2. Second, comparison of our in-house predictors to the other available tools in the field. In Figure 4a, we see a comparison of our MLR models against Envision for 12 proteins. The reason we show this comparison separately is to make a fair comparison. That is, Envision is a predictor also trained using functional assays and it predicts continuous scores. So when we compare our predictive performances, we see that more or less we are in the similar range with Envision, where for 4 to 5 cases we even outperform Envision. Then we did a comparison against 19 tools. Here, the goal was to see the predictive ability of the in silico tools per protein and make a comparison against binary models. When we look at Figure 4b, we see that our predictors are in the average range of predictive power, showing that our choice of features were good to achieve a predictive power. Also, we see that for proteins like TPK1, CALM1, for which we had the lowest Spearman rank values, other tools also give poorer performances. And lastly, some specific models work for other proteins, clearing the way for application of PSPs to clinical genome. What we see in Figure 5a is a heat map of Spearman's ranks, where rows are the trained protein-specific models and columns are the predicted proteins. This initial analysis showed us that there are some models that work well for other protein assay predictions. From here, we selected those PSPs that give the highest score for a tested protein and compared them to Envision again. Surprisingly, the predictive power of these tools were also very similar to what we observed in protein-specific tools themselves, meaning that there is a possibility to use a PSP for predicting values in other proteins. So in conclusion, we developed 29 protein-specific predictors to predict the functional assay values. Our results show that our choice of features are well chosen to perform a moderately good predictive ability. Our next step is to focus on especially those proteins with poorer performance and to see if the performances can be improved by addition of other features. Lastly, with cross-prediction analysis, we observed that there is a possibility of advancing the development of PSPs by creating a mapping between the predictor and protein spaces. Thank you for listening.